and physical principles are expressed in biology. So that's how I designed this talk to include 4 self-standing but connected stories. And let's hope I won't run out of time, but I'm really eager to present this to you. So I will start with a little preamble. It was year 2013, and me and my mentee, who was also trained as a clinical dentist, we were preparing an experiment at the Weizmann Institute in Israel. And the point of the experiment was to dissect a certain amount of trabecular bone from the mandible, from the lower jaw, and to study how the structure of bone responds to certain loading conditions. So we knew that the mandible is full of trabecular bone because we got our training in human anatomy. But we were a bit greedy, and we decided to go for a large specimen, for the pig mandible. And the ratio of sizes is approximately, as you can see now on the screen. So we cut the mandible in 2 halves to put it into a scanner. And to our big disappointment, we found that inside there is no trabecular bone. It was a hollow tube. So that was quite shocking, because we expected to find certain type of a tissue, but instead it looked like a long bone, like a femur or so. And from that structure actually extended from the level of the incisors of the pig towards the angle of the mandible. And according to the simple rules of mechanics, this geometry would be optimally designed for bending, right? And we were puzzled, but then the question was answered by the internet, because I found that what pigs do with their mandibles, they dig. They search for roots or truffles, and they use their incisors as a spade to lift up soil. That's why the mandible is in general loaded in bending, not in compression as we expected. But then that's clear for tubular structures, for long bones, for compact bone, but what is trabecular bone for? And trabecular bone is located at the sites of the skeleton where bones connect to each other, in the joints or in the vertebrae, and these are the places where movement is possible. So we have this very intricate tissue, which is a 3-dimensional network of tiny struts and plates. And these trabecular elements, of course, they line up according to certain stress trajectories, but they're also enclosed in a very thin cortical shell where bones contact with each other. And the question that intrigued me for a long time was, what is the point? Why nature puts such a delicate structure that can be easily broken on a dry specimen in the heavy-duty locations such as the spine or the hip or the knee? So this brings me to my story number 1, the topology of trabecular network. So if we produce a 3-dimensional image of a bone specimen that would be in fact full of trabecular bone, and we have enough material to study, then we can simplify this structure by extracting the network parameters from a 3-dimensional tissue sample. So let's say every trabecular strut will be replaced by its simplified version, an edge, where 2 or more or 3 more edges connect in space. We will have a node. So depending on how large is the specimen that we're interested in studying, we will produce a certain population of nodes that are connected at edges. It's a very abstract mathematical presentation of the network, and every node has 3 coordinates in space, which are unique. And every edge would have also 2 sets of coordinates of its origin and the terminus. Now, when we know the coordinates, we can apply certain rules of vector calculus to compute what is the average angle where several edges connect at a node, what is the valence of the node, and what is the distribution. So we run that study. And theoretically, in fact, one can span 3-dimensional space only with nodes of 4 neighbors, nodes connected to 4 other nodes. And that would be the classic diamond lattice. But in bone, actually, the situation is way more complex. The majority of the nodes that comprise the lattice, the network, they are very simple ones. They're the nodes of 3 neighbors. Less than a quarter of the nodes are those of 4 neighbors. And then as the node coordination or valence increases, the abundance of that type of a node drops down. Now, for every given configuration of a node, there is a basic rule that the edges emanating from that node would be dispersed, would be spread in space to cover maximal area. So for a node of 3 edges, that would be a Mercedes-Benz type of structure with the average angle around 120 degrees. For a 4 neighbor node, we will have 109, which is the tetrahedral angle. And then for more rare nodes, the average angle will just go down. We interpreted this 3-dimensional assembly of trabeculae in spongy bone as an adaptation, first of all, by having this minimal network complexity to provide certain resilience and bouncing property, because where trabecular bone is located in joints or in the spine, we cannot actually put cartilage between the rock and the hard place. And it's important to have certain shock attenuating properties of bone tissue. That's why the connectedness was so scarce. And the interpretation of having this maximal spanning of 3-dimensional volume was that it's important for movement, regardless of the direction of the impact that comes to this anatomical area. There will be always a subpopulation of trabecular elements that would meet this impact heads-on. And then we repeated the study on several species, different joints, different areas within the same joint, on humans, pigs, sheep, and we found the same topological pattern. The texture of bone is always different because it reflects the local adaptation. So elements could be thick or thin, aligned or chaotic, but the network parameters are the same. And then I moved to the UK, and then I had the privilege to work with historical collections in pathology museums, and I was able to scan the specimens from the 19th century of cultural deformation of the foot. These were specimens imported from China during the Opium Wars, and this is evidence of old cultural practice where the growth of the foot was restrained by bandages, and that was seen like in medieval China as a sign of aristocratic refinement and beauty. In fact, they affected into because the shock absorbing locomotory pattern of the foot was violated. And we could see this poor function in the amount of bone and bone density, but the network properties were still conserved. And that brings up an important question. Should we study it at all? Because it's always the same. And indeed, although the topological blueprint is conserved in species, sites, and between individuals, we do observe certain very marginal variation. First of all, in fetal bone, when bone is just forming in excess to be pruned later when it enters functional loading scheme, and that overconnected network is expensive metabolically, but this is a natural strategy to provide larger degrees of freedom to be collapsed later. And another scenario where the topological network is different is in the case of frailty and old age, where the simplest nodes dominate and the network becomes just too compliant to withstand trivial loads, and then a fragility fracture can occur. So the point here is that we have this very fundamental topological blueprint that is established early and deteriorates late, if at all. And on top of this basic structure, bone can adapt by building thicker elements, thinner elements, coarser elements, finer elements, and then the cycles of morphological adaptation can come over and again during one's lifetime. But everything is based on the same topological blueprint. So to put it in a figurative way, let's say we have different, let's say, chairs that are designed for each of them for a particular function, but they all share the same topological blueprint, such as having the seat, having the back, having 4 legs. And if we distort this topology, there won't be any function possible at all, regardless how thick or thin the elements are. And that was quite intuitive. And then we came up with an idea. Can we just scrap everything that we learned from bone topological optimization and design an alternative way of putting things together in 3D and see what can come out of that? So this is how I started my very recent project with a PhD student from your faculty, Amar. We decided to reconstruct the way bone adapts to its mechanical function. So we scanned a few bones and reproduced this topological network structure, and then extracted the nodes from this sample, and then reproduced the population of nodes following its probability distribution function. So in the natural population of nodes and in the artificial population of nodes, we would have same likelihood to find a certain number of neighbors within a given radius with respect to each and every node. Then we overconnected these nodes with a higher number of edges. So the biomimetic synthetic lattice would have exactly the same number of nodes, but the synthetic lattice would have 5 times more edges. And then this lattice was run through iterative optimization cycles of or in 3 directions. And every time, after every cycle of simulated loading, we would detect the edges that were least loaded and prune them. In the process, along this trajectory of optimization, we were recording certain preset outputs that we call generation 1, 2, and 3. Generation 1, the first evolved lattice, would be the one having minimal von Mises stresses that would mean, in like simple interpretation, that it would be the elements would be loaded mostly in stretch-dominated way. The second convergence criterion would be having an equal nominal weight of the evolved lattice and the reference lattice. And the third convergence criteria of generation 3 would be having exactly the same number of elements as in the reference lattice. So we had, therefore, 5 objects to compare. And for each of them, we were also monitoring their topological descriptors, such as the proportions of the nodes of different valence. And when we compared them, the mechanical properties and the modulus and the distribution of stresses within these lattices, we found that when optimization was done in 3 axes, the biomimetic lattices, when having the same mass as the reference, they had 10 times higher bulk modulus. They had significantly more prevalent axial loading, and the distribution of stresses, as we see from the standard deviation of the local von Mises stress, was very uniform to compare to bone. And even we reduced the mass below that of the reference lattice and arrived to generation 3 with the same number of elements, we still had superior mechanical properties in comparison to bone. So that was very encouraging, because it means that, first of all, if we know the principles of natural design, we can stretch it a bit further and maybe outperform or just adapt it for whatever engineering reason we have in mind. It is also possible to use the same optimization recipe, the same generic trajectory of optimization for achieving various desired properties. Let's say, for example, I want to obtain a structure of minimal weight, and that would be band dominated for a sacrificial structure. And I can do so just by running the optimization loop a few more cycles further, and then recording the lattice, which would be very lightweight and would have a certain proportion of bending and stretching in the elements. And the last important conclusion from that study is that it is possible by careful design and bioinspired optimization to minimize the amount of material we use for fabrication. And this is important, especially today at the time of ecological fragility of our planet. So, and from here, I'm going to zoom in and now present another part of my study of order and chaos in our skeletons. So this pertains to the scale of micrometers. So this story is a bit older than the previous one. It started in the beginning of my PhD, where my advisor, Steve Weiner, put this paper on my desk, and that was a review of the hierarchical structure of bone. And I was given a task to replace the cartoon with the real micrograph. So this schematic shows that bone has quite a complicated hierarchical assembly of structures, nested structures, starting from the crude components of mineral and protein called collagen. Mineral resides and is associated with collagen to form mineralized collagen fibrils. These fibrils form registered arrays. And these arrays, depending on the tissue purpose, form different patterns of various degree of anisotropy. Then what we know mostly from light microscopy or some simple electron microscopy observations, and we can find concentric cylindrical motifs in bone called osteons, then spongy or trabecular bone and compact bone, and at the highest level, entire skeleton element. So that was my task, to make a real image. And of all these types of tissues, the most common one is this one in the yellow frame, which is so-called lamellar bone. As the name implies, lamellar bone has a plywood-like structure. So if you break a specimen and look at the fracture surface using an electron microscope, we will see that indeed collagen fibrils, mineralized fibrils, that stick out of this fractured surface at different angles. And the structure repeats itself with a period of about 3–5 microns or so. There are also cells residing in this extracellular matrix, and they communicate with each other through, so here would be the cell, and the communicating channels would be, if you can see my arrow going through the mineralized matrix. But this is a fracture surface, right? It doesn't give us complete high fidelity three-dimensional information. And exactly around that time, the dual beam microscope, also known as focused ion beam or scanning electron microscope, made its way into life sciences. And that was quite a serendipity, because I could modify the method of operation and preparing the specimen for getting true three-dimensional information about bone organization. The principle that works is that the microscope has two guns. One is the ion beam, which operates as a very fine knife, and it is capable of ablating a layer from the specimen surface. And the other gun is a conventional electron beam that rasters the surface and produces a backscattered electron image. And these two beams are oriented at an angle with respect to each other, and by iterating their activity, we can acquire a set of two-dimensional pictures. When the acquisition is complete, we can stitch them digitally to form a true three-dimensional image. Because of the angle between the two beams, the geometry is not exactly orthogonal, it's rather a diamond shape. And these experiments are usually very long. So here I have two asterisks that indicate the moments where I came back to the facility next morning to adjust the field of view, and then digitally align these areas and stitch them together. So the beauty of the method is that after the images are all put together and aligned, we can reslice it in any arbitrary plane and just observe any possible feature. Of course, the most salient feature that we can segment and visualize would be the cellular body. That's easy to process. But my objective was to study not the cells, but the extracellular matrix, which is around the cell. So now I'm going to play a few videos, each of which makes this story complete. So here, this plane, this grayscale image will be moving bodily to the back of the screen, and that's actually the sectioning plane. So it will show how collagen fibrils are aligned in 3 dimensions. So we begin with this orientation from the top left to the bottom right, and we can recognize collagen by having this characteristic banding pattern. So let's watch. First, we start with quite aligned collagen, forming sort of linear arrays, and then it loses its orientation and becomes chaotic. And then they regain their degree of alignment, this time vertically, and then there is another spacer of chaos. And then the structure repeats itself. So we have aligned, chaotic, aligned, chaotic, aligned, chaotic. Now, because it's a true 3-dimensional image, we can tilt it and look from the other projection, and then we see that the horizontal fibrils form sort of cylindrical bundles that are organized into a monolayer about 2 or 3 microns thick. And if now we tilt and flip this 3-dimensional image in another direction, we will see that the high angle packed high order fibrils also form a sort of an array of bundles that are co-aligned. And at the interfaces between the layer of bundles oriented one way and another way, we have these spacers of chaotically organized, disordered collagen. Here is another specimen, another donor, another type of bone, and we see the same organization plan. We have aligned collagen packed into bundles, then there is a layer of chaos, and then there is another set of bundles oriented in different orientation, and then the structure repeats itself over and over. In this one, in this specimen, which is again a different type of bone, this time it's animal bone, here we can see cellular processes that percolate through the mineralized matrix to make connection with another cell sitting at around 20 microns distance. And here it's quite obvious that cellular processes that are delicate, they penetrate this organized and aligned matrix through the chaotic elements, just because they can. This is the only possibility to percolate through densely packed collagenous material. And the hubs and connections between individual processes are confined to the chaotic layers of collagen. So, and this picture is a figure from a paper that I will now just tone down a bit. It shows the relationship between order and chaos in 6 specimens of human bone. So here the black graph stands for the direction of ordered collagen fibrils, and the gray graph stands for the extent of chaos. So where the chaos goes up, the direction changes. Now, when I speak about the bundles that are all co-aligned and they have quite a nice registry of collagen fibrils, in fact, within every layer of bundles, we see a very subtle, very delicate change of directions, which means that monotonous orientation of fibrils in these bundles is not the case. In fact, they are slightly twisted as loose ropes or like braided hair. And back then, we designed a new schematic for a lamellar bone, the most common bone of skeletons of large mammals, as indeed a plywood-like structure that was in fact composed of ordered and disordered materials that alternate. The ordered collagen fibrils packed into bundles that have a very low-pitched twist, and the chaotic phase engulfing the entire cellular network. And my estimate back then was that the chaotic phase, which is here depicted in blue, comprises about 15% of the net volume of bone structure. And just recently this year, by using deep learning algorithms for image segmentation, I managed to segment actually the order and chaos from each other. And I found that chaos in fact comprises up to 25 or 30% of lamellar bone makeup. And to my greatest delight, it was the first time when I could, by reprocessing these old scans, I could visualize the twist of ordered collagen bundles. So now I will switch to another scale of magnification, the scale of nanometers, and then make my way back to the level of skeletons. It happened to me to watch a very nice series, BBC program, The Secrets of Bones, where the operator took two skulls of small animals and he treated them differently. One skull was soaked in acid to demineralize, and the other skull was incinerated in the furnace. They, in the end, behaved absolutely differently mechanically. So the demineralized skull was like a bouncy rubbery ball, and the deproteinized skull was very fragile. But regardless of this mechanical behavior, the shape and the size of bone was exactly the same as in the original specimen. So we know about collagen that this is because of this interspersed phases and branching and twisting bundles and the chaos and the cells, but what keeps the mineral phase together? So that was quite a long debate in the history of bone research. It wasn't clear whether these crystallets of hydroxyapatite in bone are needle-shaped or platelet-shaped, whether they are inside collagen fibrils or outside. And by just making more and more transmission electron microscopy specimens and producing them in large amounts and observing them, all of a sudden, I managed to see, instead of 2 projections of mineralized collagen fibrils, 3. And depending on how you view the orientation of mineral and collagen in bone, it appears in 3 different fashions. When the fibrils are oriented more or less with the plane of the image, they look like wavy formations, like braided hair, with a certain curvature with a radius of around 200 nanometers, separated from each other by uniform repetitive gaps of around 1.5 nanometer or so. When the section is oriented oblique, then we still have curved crystals that are separated from each other by these uniform gaps of around 1.5 nanometer. And only when we are lucky enough, in very rare case, when we manage to cut mineralized collagen fibrils exactly perpendicular to their axis, they are oriented in these galaxy-like assemblies, like spiraling formations, each of which has the maximal radius of around 100 nanometers. And we can also see crystallographic reflections that tell us that these crystals have hexagonal symmetry. And the fact that instead of having 2 views of the same structure, like on face and profile, we obtained 3 views of the same thing. The only model that could explain the coexistence of 3 motifs of mineral packing in bone would be the model of superhelical assembly of very thin and merging crystalline particles. And we call that cross-fibrillar mineralization model. Interestingly, at this level of magnification, with the resolution of around 1 nanometer, we can see that mineral particles have their own extent of hierarchical structure. So firstly, they form needle-like crystals, like the fingers of my hand. And then they merge to form like the palm of the hand, and several crystalline particles of that geometry form stacks that are separated from each other by very uniform gaps. And at the level of these hands or platy crystalline assemblies, they exhibit a bit twisted, gently twisted geometry, like a blades of a propeller. And actually that resembles quite closely what we see 2 orders of magnitude coarser than that. So now if we connect the dots and all the information that we know from gross anatomy and histology and electron microscopy studies, an interesting picture will emerge. So at the level of gross anatomy, bones have a certain twisting pitch. The ridges are twisted. And then within every bone shaft, we have capillaries that propagate through the cortex following a very gentle 15 degrees twisting pitch. These are Hierarchical canals. Around each of them, we have an assembly of lamellae, and every lamellar structure is twisting around the central canal with a varying angle. Every lamella is made of bundles, and bundles are twisted as loose ropes. And every bundle is made of mineralized collagen fibrils that is the basic building block of the structure, and it's a composite structure that is made of superhelical microfibrils of collagen that are made of triple helices that are made of helices, and it also incorporates these cross-fibril or curved crystals that display a propeller blade-like twist. So essentially, this self-similarity that we observe at least at 9 levels of bone structure tells us that bones are fractals, which is a very common theme in nature that biological design uses and reuses in organisms. And now I'm going to switch to the last part of my research talk, something that I have accomplished recently as an application specialist with the IT company. So I have shown this picture to you, and I made the point that depending on where bone is located, it adopts different morphological characteristics. So if this bone is loaded monotonously, it will have a very oriented anisotropic texture. If it has a remnant of growth plate, it will have a solid inclusion. So, and so on. So sometimes when we step just a few millimeters aside within the same specimen, we may discover absolutely different morphological fabrics. And one important parameter that is classically used in skeletal research is the degree of anisotropy. There are some classical methods that are applied to compute anisotropy, such as mean intercept length or star tensor. And all these methods are statistical. So you need a certain amount of volume to draw these virtual lines, to compute their segments length, to build a tensor. And for that reason, it's of quite low resolution, because then this entire proximal femur of a human would have certain anisotropy value, let's say 0.63, which doesn't tell me what does it relate to, which aspects of the loading history of this specimen it reflects. So we decided to build our own anisotropy measurement algorithm that would be based on the co-alignment of surfaces and would result in a map, in a 3-dimensional map of varying anisotropic features. So this is a simple body on the screen, this box, that let's say we divide its surface into certain units and construct a vector perpendicular to every unit of surface. So the top surface, which is the smallest one, will have just 2 vectors, and this the surface pointing to the right will have 10 and then 20. And if we make an inverse and reciprocal, we can say that the structure is indeed elongated vertically, and it's anisotropic. So we could do the same for micro CT scan of bone, for example, by constructing a surface and building these units of the vectors. And by applying this algorithm to an entire bone specimen, we can construct exceptionally rich maps that tell us the story of what does this bone do at every point of its structural organization. For example, this is a proximal femur of a sheep, and here we can plot anisotropy in terms of its magnitude. So low anisotropy, blue, high, and red. And it shows that the low anisotropical features are located where this bone contacts another bone, the pelvis, or where the muscles get attached. But besides plotting it from low to high values, we can also plot it in terms of orientation, borrowing the crystallographic notion of red, green, blue standing for X, Y, and Z. And that is really curious because here in this proximal femur, we have certain features that are expected to have low anisotropy and diverse orientation, such as in the femoral head. But this area, the trochanter, serves as an attachment point to very strong muscles that control the posture. And this is the minor trochanter that serves as an attachment point to another group of muscles. So we have a pulling vector here, and it means that this tract of co-aligned trabeculae works in tension, which is, for a bone biologist, is quite counterintuitive and exciting. And the same feature we can see on the other extremity of the same bone in the knee. We also can identify structures that apparently, according to their geometry, are loaded in tension. So to conclude, my view of skeletal adaptation is that function is first, and it molds the structure. That limited inventory that is at disposal of nature is not an obstacle for maximizing diversity. And one can build a panoply of different, perfect, elegant, effective structures just by combining order and chaos and hierarchical assemblies and self-similarity. And I think my most favorite conclusion that I feel comfortable making is that biology is an exact science. And we are now at the point where it's about the end of observational studies. Now we can apply notions of physics, mechanics, and mathematics and expand our understanding of nature. I would like to thank all these people who helped me starting from my PhD and my 2 postdoctoral appointments and during my industrial experience, and the funders, the Israeli Science Foundation, the Wellcome Trust, and 2 NSERC Engage grants that funded my postdoc in Canada. And if you allow me to present just 6 slides that address my future research, I would proceed. So the first project that I have in mind refers to structural water. It's a common knowledge that a human body contains about 60% of water, but the skeleton alone is made of water by 30%. Most of this water is in interfaces and in the marrow, and if you get rid of that free water, we will still end up with about 10% of water content. This 10% sums up to 1 liter of bound water. By bound water, I mean that this water is strongly associated with other components, with surfaces. And one peculiarity of bound water is that you cannot extract it unless you heat your structure beyond the boiling point. So as this thermogravimetric graph shows, that a specimen of bone loses 10% of its mass when heated between 100 and 200 degrees. So this is bound water, its contribution to the composition. Bound water is associated with mineral crystalloids that I showed are very fine and fractal-like. And every charged element of the crystalline lattice carries quite a significant thickness of structured rigid or water dipoles. And because of the number, the sheer number of these crystallines that are associated with water, if we calculate the collective surface of all the components of bone, we will come up with a terrific number of 0.9 kilometers square, which is about the downtown New campus area times 3. And if you take 1 liter of bound water and distribute it evenly over this enormous area, we will get a hydration layer of about 1.5 nanometers, which is quite in agreement with the simulated amount of structured water that one can find on the surface, and all that within the same body. Now, what does bound water do there? And it actually becomes quite apparent if we look into certain methodological phenomena. So one way to study bone is to produce thin electron transparent specimens by upgrading bone sample with an ion beam. So we do that by using unmodified natural bone. We put it into an electron microscope, produce a thin slab of 1 micron thick, and we start thinning it with a focused ion beam. During this process, to constant disappointment of the operator, these specimens warp. So let's say I'm making a specimen about 7/4 microns in area, and when I thin it in high vacuum beyond 200 nanometers, it starts warping to, well, up to 1 micron deflection, sometimes less. So what is happening there when nothing is directly acting on the specimen, it's bound water leaving the surface of the specimen and vanishing somewhere in the pump of the microscope. And by doing so, internal pre-stress is released and the specimen warps. And for being fixed on both ends, which is the case of this electron microscope specimen, we have a formula that allows us to compute what is the force acting on the object. So if we substitute some known values, such as the deflection, the extent of warping, the dimensions of the beam, its second moment of area, and Young modulus, it actually results in this amount of force acting perpendicular to the lamella, and normalized by the area, it actually puts the pre-stress value in the ballpark of air pressure in the tires of a motorized vehicle. And this pre-stress is mediated by water. So the more water in the tissue, the more pre-stress is exerted on other structural components. And the water binding capacity of extracellular matrix is defined by the pH of interstitial fluid. The lower the pH, the less water binding capacity can be observed. Now, pH in a living organism is very strictly controlled, for example, in blood, because blood has a huge buffering capacity. But in the interstitial fluid, we have quite a huge lag in adjustment of pH. And there are certain conditions that acidify the environment, such as inflammation or metabolic diseases or aging or just death. And other processes actually raise the pH towards more alkaline values. And this is, for example, strenuous exercise, aerobic exercise. So my plan is to, I don't know if you can see it behind the thumbnails, but to run this study of bone crystal morphology in low temperature condition, in cryogenic conditions. And I would like to study that using an animal model of acidosis and compare it with a wild type animal and the animal subjected to strenuous exercise. And that is quite a massive project to be submitted to CIHR. But before I do so, I have to fix my syntax, right? To develop the workflow, how can I measure and calibrate the amount of pre-stress in bone? And that should account for the way collagen is assembled, how it is cross-linked to make it inextensible, what is the osmotic agent that is enclosed in the assembly, and what is the ambient pH. And then it's important to do that in a cell-free environment so that it will be more controllable. And we can monitor these properties by micromechanical testing, measuring amount of water by spectroscopical method, or by just thermogravimetric studies. And there is a system developed by Shoan Nazat in your faculty that allows to assemble collagen with a high degree of precision and desired alignment. And it also allows for mixing in other components, other dopants, such as polyanions, and to cross-link the system. So this is something that I would be interested to pursue as a pilot study to be submitted to NSERC. Now, I also, if I may, I would like to show another project that is more of a biomedical character. It's also bioinspired. So this structure on the left is the vertebra of the arm of a brittle star. It has the same topology, surprisingly, as trabecular bone, but it also incorporates a gradient of feature frequency. So depending on what is the function of this vertebra locally, the pores can be larger or smaller, or the structure could be nearly solid. I have at the moment a recipe for optimization for designing of topologically optimized assemblies. And also I have done quite a massive study in the UK on using 3D printed polymer scaffolds for bone regeneration. And I know that when produced by selective laser sintering of polyamide, these scaffolds can trick bone cells into thinking that this is a natural bone surface, and what cells do, they just continue building new material, new bone on the surface. And these three important things actually point to quite an obvious application. We can 3D print a scaffold for reconstruction in any customizable shape. Polyamide is a very convenient material because it's bioinert, and it also can be adjusted intraoperatively. And the polymer nature of these scaffolds would allow adding another level of reconstruction, such as dental implants or ocular implants and so on. And this is of high advantage in craniofacial applications where precision is the key. And usually reconstructive surgeries are multi-stage procedures that last for several years. So I would like to pursue the refinement of topological design by incorporating gradients via NSERC Alliance funding, and then proceed to clinical trials with colleagues from orofacial surgery. And I would also like to develop, I will be really brief now, I would like to develop an application for the anisotropy mapping algorithm as a way to monitor the effect of lifestyle and physical activity on the textural adaptation of bone in human knees as a joint project with the skeletal collection of the University of Tennessee. And that will be submitted to CIHR. So that's all. Thank you for your attention.
Thank you so much, Dr. Reznikov. Dan,
I guess that it is the clapping.
Exactly, yeah. we can use the chat option to just send the clapping emoji, I guess. Dan, would you prefer for us to move into the teaching seminar or have a quick Q&A right now?
No. Let's have the questions now.
All right, sounds good. So for everyone, if you want to ask a question, please turn on your cameras, wave at me, and then I can say that you can be unmuted, or type in your questions into the chat, whichever you prefer.
I have a very simple question. First, I am a microscopist. I am in material engineering, and I have to say that the microscopy work is spectacular. The FIB work is first class. But I just have a very technical question. You mentioned that it's fractal over the length of scales. So have you measured the fractal dimension?
Yeah, I measured. So it's about 2.6. But I'm not sure that the method I used is appropriate for every size scale. So it would be really amazing to adjust the algorithm to be appropriate for every scale of resolution.
Okay, so what technique have you used? Have you used a frequency technique or the mass dimension? Because I think with the 3D data, the mass dimension should be easy.
I used a plugin in a very common software called ImageJ. It's fractal dimension, so it kind of splits the 3D volume into units of volume further and further, and then monitors how.
It's the box dimension.
Yes, right. Yes.
But you should try the mass dimension, I think. So, and the beauty of the mass dimension is you can see change with the length. So there is what they call the percolation length.
Okay.
And probably the percolation length will be related to the size of your cells, basically. So it would be interesting. Anyway, that's a technical detail. I think I did enjoy very much your presentation. Thank you.
Thank you.
Right. Thank you very much for your question. Next in the queue is Professor Erlicher. I've unmuted you, Ellen. Go ahead.
Okay, great. So thanks for the talk. I found it really interesting, fantastic work, and some things I'm very interested in. Let me just ask you a first question related to the previous one about the fractal dimension. Do you see any variation of this on different scales related to the local stresses or strains that the bone is subjected to? Or is that more of a scale of the actual structure that you're seeing vary?
Well, there are many unanswered questions yet for me in person. For example, I think that when I will finally image the crystalline morphology in cryo conditions, I may find a steeper twist because the pre-stress would be modified in a different way. Probably it will be more pristine if I freeze the samples and image them in cryogenic conditions. Mm-hmm. Then I also think that this is a generic solution of nature, and it would not reflect individual adaptation and loading history. But it's a very rich area to just go ahead and probe. Maybe different diseases would result in violation of this self-similarity rule at various scales, but it can be also compensated at one hierarchical level or another. So I don't have a very simple question answer to that.
Okay. Oh, that's fine. One other question, if I could, and then there's a lot more we can talk about this afternoon. So I was really excited to see your different kind of phases of, you know, ordered structure and then chaos, and then order and then chaos. And I think one of the design features that's ubiquitous for these asymmetric composites like bone or nacre, you know, things like that, are these combined stiff inorganic and then compliant inorganic phases. And I was wondering if you can relate those phases of order and chaos to the relative toughness of the material.
Okay, I think that because cells sit exclusively in the chaotic felt work, because it's more accommodating, they are located there because it's also a bit more compliant. We did a little side project on simulation, and we found that it responds with a larger strains for gross deformation of the specimen. I think this is a way for the cells, for the strain gauging element in living tissue, to sense dangerous, detrimental stresses early before it deteriorates the entire structure, and then to trigger appropriate adaptational mechanisms such as resorption and apposition, building up more bone, and so on. Yeah. And also, it's kind of it's the thing of nature's toolbox. Minimal inventory, maximal diversity. In nacre, in timber, in bone, there is the same thing. You have high stiffness, low stiffness elements, high anisotropy, low anisotropy. So it's just a generic recipe.
Okay. Great, thank you very much. I look forward to talking to you this afternoon.
Thanks, Alan. If anyone else has more questions, you can type into the chat so I can unmute you, or you can just wave at me.
Hello.
Adam.
Adam? Okay.
Adam, unmute yourself. Oh, Luisa does it. Okay.
Okay, should be good.
There we go.
Hi.
Hi. Yeah, I thought it was a really interesting presentation. I had a question in your proposal to plunge freeze large bone samples and then do milling of the frozen samples. I'm wondering about what challenges you would foresee in maintaining vitreous ice with these thicker samples and avoiding any sort of crystalline formation or anything like that with the water in that preparation.
Well, there are several ways to freeze a sample. If the sample is in the order of magnitude of up to 10 microns, we can use plunge freezing. If it is up to 200 microns thick, we can use high pressure freezing. High pressure freezing combines just changing the phase transition diagram of water, and therefore is more permissive in terms of sample thickness. We cannot reliably vitrify water in a specimen thicker than 200 microns without using a cryoprotectant. My plan is to prepare the specimen for freezing when it is already thinned to, let's say, 2 microns, where it is still not sensitive to this reduction of surface to volume ratio and will still keep its structural water inside, but it can be already frozen with a high certainty of vitrification. Now, with plunge freezing, in fact, there is an imminent threat of distorting it because of the shear force acting at the moment of plunging. So perhaps it's more conservative to freeze it using high pressure freezer, which would be just more gentle. And then it's actually not a very complicated procedure to position it on the cryo stage and to mill it with the focused ion beam and to see what will happen. But nobody has done that before. So I guess there is about a year of optimization to run and to come up with a good protocol.
Any further questions?
So if we have none, we can go to teaching.
Okay.
I guess many of us will meet Natalie today, but a couple of us. All right.
All right, so I'm launching the teaching talk. Okay, so it's intended for students, but I want to make an announcement to you. There are certain topics in biology that I find exceptionally fascinating and also fun, such as something that we know intuitively from everyday life, the nonlinearity of biological systems, the graded interphases and interfaces, hierarchical materials, sacrificial structures and self-repair, bound water, obviously, pattern formations, and closed kinematic chains, for example. And I never can help myself not to mention at least one of them in any of my bone talks. And it just occurred to me that the philosophy of your department, the framework of bioengineering, is the ideal place to just put them all together and to design a very enjoyable, eye-opening course that would be a very nice introductory level undergraduate scenario for young students. And today, out of these modules, I would like to focus on module number 9, closed kinematic chains, in the light of which I will talk about the biomechanics of joints, and I will speculate a bit and propose a solution to a certain clinical problem that figuratively, we can talk about problems and their solutions as of a landscape where we know certain features and we know our path and we are trying to find a solution to a problem. In the context of mechanics of movement, there has been a historical separation between artificial man-made structures or human-made structures, to sound more modern, and natural animal structures. Traditionally, we look at artificial structures from the point of view of Newtonian mechanics and lever-based mechanics, whereas living organisms rather employ closed kinematic chains in their movement. The point here is that these two theories, these two schools of thought, do not contradict each other. They actually overlap, and it's just a matter of expanding the illumination spot for better understanding of certain mechanical phenomena. So the learning objectives of this presentation is to present closed kinematic chains, how they're made, what do they do, what are their main attributes, how can we identify a closed kinematic chain in a living organism, how these can be applied to one particular joint of the human body, the temporomandibular joint that connects the mandible to the skull. And then I will express my concerns about a very common dysfunction of a joint in humans. And I would like to propose this study for the bioengineering design project. So a closed kinematic chain is an assembly of rigid bodies that are connected by joints to each other. The difference between open chains and closed chains is purely topological, right? The difference also is more pronounced when the rank of the chain is low. So for example, here we have an open chain of 3 links and it's very floppy, and the closed chain of 3 links is absolutely rigid. Then the difference is a bit more vague for a 4-link chain, where the tetragon closed chain can collapse into a rhombus, and then if we increase the complexity of a chain, then the difference becomes really vague, and for a 50-link chain, it doesn't matter whether it's closed or open. But we can mimic an open chain by using a closed chain. For example, here a 6-link chain is folded on itself, and it looks morphologically as a 3-link open kinematic chain. Now, at every this transition can be done only in one direction, right? We cannot mimic a closed kinematic chain by an open chain without closing it. Now, at a particular level, at the relationship between 2 adjacent links and their joint, the rules of lever mechanics apply perfectly. But in the case of closed kinematic chain, the relations between one set of links and a neighboring set of links and remote set of links can influence each other. So while locally the rules of lever mechanics apply, globally it behaves rather like an assembly of logic gates. Here's an example where we can mix up, is it a closed chain or an open chain? So at the first glance, a human digit appears like an open kinematic chain with the free end moving in 3 dimensions. But if we look closer at the anatomical assembly of tissues and ligaments and muscles, it is in fact a closed kinematic chain where we have, for example, in a very simplified way, we have 2 major tendons that are attached to the muscles of the forearm. There are no muscles in the digit. So, and these 2 tendons, by being pulled simultaneously or not at all, or just one or the other, they can perfectly control the position of the fingertip in 3 dimensions. I would like to play this movie that shows an artificial structure, strand beast, that is walking, being propelled by the wind, and it moves surprisingly in a smooth and lifelike manner. Moreover, some legs of this creature are redundant, and if one of them is getting stuck, like at this moment, the other legs continue the movement without any interruption. The superstructure of this creature is very stable, so you can put any piece of equipment or a sensor or a brain on top of it, and it will be functioning undisturbed because of this very smooth movement. The basic element of closed kinematic chains, and that is also the foundation of this strand beast that is capable of lifelike movement, is this simplest flexible link known as the pantograph. The pantograph is an old invention of artists that was used originally to reproduce certain shapes with a high degree of precision. Here, by modifying the length of the arms and by applying some force here, we can either attenuate or augment the amplitude of the movement. Interestingly, because it's a very simple chain of just 4 links, the trajectory of the input and the output is the same. But now let's dissect the leg of the strand beast and see which elements constitute this basic feature. So, first of all, this is a more complex kinematic chain. It has 4 basic elements, of which 2 are rigid. These are the triangles A and B, and 2 more elements are collapsible. These are the tetragons N and M. We also have the element to which force is applied. This is the actuator, and we have the effector that interacts directly with the substrate, with the environment. Now, let's split the cycle, one stride of the strand beast, into individual phases. So here we apply a rotary movement to the actuator, and that displaces the entire assembly of the leg, its center of mass, forward and upwards. In this process, the top tetragon opens up and the triangles displace bodily. As the movement is continued, then the top tetragon collapses, the second one opens up, the triangles continue their displacement, and the stride is accomplished. The effector shifts in space in the direction of propagation. And then the structure, the movement repeats itself cyclically. So I just drew this schematic in PowerPoint, but there is a paper by Jansson and colleagues who simulated this movement in very fine increments. And when they superimposed these individual increments, they found that not only the amplitude and the force change by the operation of a closed kinematic chain, but also the trajectory of the movement of the effector is different from that of the actuator. And that is thanks to having these rigid shapes that affect the trajectory of displacement in a certain predictable and smooth manner. Zoologists have studied closed kinematic chains for a really long time. Every animal uses closed kinematic chain primarily in its feeding apparatus because it's effective, it's energy saving, it's just cheapest metabolically. The most celebrated example, I think you are familiar with, is the Peacock Mantis shrimp. It's a marine crustacean that feeds on clams, and clams have these over-designed, very rigid, tough shells. So the Peacock Mantis shrimp uses closed kinematic chain when it deploys its club that accelerates to that extent that it produces water cavitation when it contacts the shell of the mollusk, and of course breaks the shell and allows the shrimp to feed. But natural kinematic chains, as opposed to what I just showed to you, are multi-link chains. They're rarely 3-link or 4-link. And every component of a biological kinematic chain could be either a muscle or a bone or a combination. This multi-link character of kinematic chains is inherently chaotic, right? They're unstable. And for the purpose of stabilizing and achieving high degree of control and precision of the movement, some parts of these chains have to be stabilized temporarily. Which parts of the chain become stabilized, it depends on the biomechanical task of the organism. So here, for example, if the proximal part of the chain is stabilized by triangulation and tonic contraction of the, let's say, muscles, then the distal part can perform very precise movement. Or we can do it the other way around. We can stabilize by triangulation principle the distal part of the chain, and then the movement, the effect is achieved at the proximal part. Such as in the case of these two individuals. So when Glenn Gould is playing piano, his shoulder is stabilized and the fingers can exert very agile and precise movement. And when this gentleman is climbing on the face of a rock, he does exactly the opposite. He stabilizes his fingertips and the forearm muscles, and the only free moving segment of the arm is the proximal segment, the shoulder, that exerts the force on the entire body and lifts it up. And both of them share the same anatomical plan, the same bones and the same muscles, the same innervation circuits. So this phenomenon is called heterarchy. It is the ability to propagate the effect one way or another way. Moreover, the fact that some of the links in a closed kinematic chain are muscles, and muscles can contract, is the postural effect of biological closed kinematic chain. So a certain contraction or lengthening of one link defines how all the other components of a chain will move in 3 dimensions. And for example, by having a different basic tone of abdominal muscles, an individual can control the position of the pelvis, and the position of the pelvis to a large degree defines the alignment of the knee, whether that individual would be aligned as a knock knee or bow-legged. In addition to that, closed kinematic chains in biology are not 2-dimensional, they are 3-dimensional, and they have their own nested hierarchies. So they're made of smaller chains that are put together into larger chains. And all these properties contribute to high versatility, high specialization for a given function, precision, control of movement, and adaptability to biomechanical circumstances that the organism can change on the go. There is one example that I want to bring to your attention where conventional view of lever mechanics doesn't explain certain things. This is the temporal mandibular joint. This joint is paired, it has a disc inside, so it's complicated anatomically. It is capable of producing very intricate movements, and it participates in the functions of feeding, communication, even emotional expression. And according to different population studies, up to 40% of adults in their productive years are disturbed at one moment of their life or another by the temporomandibular joint dysfunction. So when we look at this joint from the point of view of lever mechanics and consider it as a lever of third class, first of all, it's very mechanically ineffective. Well, in humans it's not so dramatic, but consider a crocodile or a cow, where the snout is long and the lever arm is long. But it is also dangerous from the anatomical perspective, because the bone that makes up the fossa, the articular pit where the mandible sits, is very thin. It's actually, here it's highlighted by the red line, it's about 200 microns. If you take a dry specimen, dry skull, and look across the source of light, this fossa will be paper thin and transparent. However, when somebody falls on the chin or gets an accident or a blow, this thing doesn't break, the mandible breaks. Otherwise, even biting on a carrot would be equal to committing suicide, because the mandible would just go straight into the brain through this paper thin layer of bone. We can also speak by having our chin, as I'm doing right now. I'm moving my head by my muscles of the back of the neck. So it doesn't line up with the theory of the lever of the third class. So the way to figure out how the mandible operates on the skull, you have to look more closely at the anatomical components. And this is my sketch of the muscles that put in movement different components of the head and the neck. If you ask me if we can simplify that, we can, but not to the degree of class 3 lever. And to make it as simple as I can, I can keep just 7 components, 7 groups of muscles that put everything in movement. And besides the mandible, very important player in this game is the hyoid bone. Hyoid bone is quite a miniature horseshoe-shaped bone that is located right below the mandible, and it is connected by different muscles to the chin, to the shoulder girdle, and to the base of the skull. In my schematic here, all these pink, orange, and red colors stand for these muscle groups being innervated by the single nerve, by the trigeminus. But when the hyoid is concerned, besides that nerve that controls the movement, we also have the contribution of the facial nerve and the hypoglossal nerve. So this little element in the middle of the neck receives innervation and control from 3 independent circuits. Now, what happens when we open the mouth? Out of many muscles that pull the mandible and the hyoid in different directions, actually few are activated. These are the protruders on the top and the mouth openers that pull the chin backwards. A couple of forces is exerted on the mandible, and obviously the temporomandibular joint is unloaded in this case. When we close the mouth, then the couple rearranges slightly, and now we have a very massive bundle of muscular tissue of the temporalis pulling the mandible up and backwards, and also we have the masseter pulling it in the closing direction. So it's a different couple, but the center of rotation around this area, and again the temporomandibular joint is unloaded. When we move the chin forward, this is a bodily displacement of the mandible, and here it is unloaded again. And the only scenario where we do load the temporomandibular joint in compression is in retraction of the mandible. It's a very painful and uncomfortable movement with the amplitude not more than 1 millimeter. It's very difficult to reproduce for a healthy person. In order to accomplish this tiny, uncomfortable movement, nearly all muscles have to fire. Primarily, the muscles that anchor the hyoid bone to the base of the skull and to the shoulder girdle. Otherwise, it would be simply closing movement. Now, why do we need this bizarre trajectory at all? That is needed for stabilization in lateral movements, in sideways movements of the mandible. So for example, we open on one side and stabilize the joint on the other side. That's the role of these anchoring muscles. But there is a situation where this very specific circuit of mandible movement is hijacked by stress reaction in modern humans. TMJ is compressed when the hyoid is pulled down. Then the depressors, the muscles that anchor the hyoid to the clavicle and the chest, they receive innervation together with the muscles that control the position of the neck. The system is hierarchical, and therefore the spastic state of the neck is associated with the forceful pull of the hyoid down and back, and therefore with the compression of the temporomandibular joint. And this is the scenario where all the features add up, explained by a closed kinematic chain. Temporomandibular joint dysfunction usually happens together with snoring and obstructive sleep apnea, which is also associated with the backward movement of the mandible and the hyoid. It is associated with the flight or fight response and chronic abnormal tone of the neck muscles. And it is also happening in similar cases with dental attrition and overloading of the bite. So it's a theory, but this is an application of the closed kinematic chain school of thought to a real life problem. Now, to make sure that everybody retained the material, I would like to ask 3 questions. Because it's a simulation, I will answer them myself. Here in this 2 pictures, we have an elephant that is breaking a tree with a trunk, and then elephant picking a fine straw from the ground. In these scenarios, which feature of a closed kinematic chain is illustrated? Hierarchy, heterarchy, or force and precision? I have a hint. It's a similar story as 2 gentlemen exerting either high power effort on the proximal part of the arm or very agile and precise movement of the distal part. This is the heterarchy. Question number 2. In this painting by Jericho, we have several individuals at different physical states. Some of them are very composed and alert, some of them are collapsing, and some are dead. This property of unconscious body languishing illustrates which property of a closed kinematic chain? It's instability of a multi-link chain when not enough stabilization between the segments is accomplished, or the integrity of a closed kinematic chain being either upside down or downside up, or maybe it illustrates the infinite degrees of freedom of a human body as a closed kinematic chain. The correct answer is 1. And the last question is, the classic biomechanics theory based on Alfonso Borelli and lever theory is irrelevant for us, or it's just accurate but incomplete and requires just expanding the research toolkit? And the correct answer, in my view, is number 2. It's just incomplete. To summarize, closed kinematic chains are common in nature. They're robust and adaptable and also allow instantaneous change in force, power, agility, and amplitude of movement. They allow us to deploy rich repertoire of control mechanisms, and understanding of closed kinematic chains enables us to find bioinspired solutions to real-world problems. Going back to my metaphor of the landscape and having different ways of seeing problems, adding closed kinematic chains theory to lever mechanics theory helps to find a way between the problem and the solution. And I have a project proposal to construct a lever-based high-frequency oscillating device that would exert vibration to the points of muscle attachment to the bones, and by this way allowing us to control abnormal tone of the neck muscles, and probably offering a solution for common dysfunction of temporal mandibular joint.
It should have been organized by coordinator as a creative framework. Because the study outcome of coordinators really requires a larger contribution because there is no proposal to invite a broad collaboration in concepts. One responsibility which have the policy that the capacity transform, which is not at all is the style of funding.